Okay, so today we are going with a topic which is the central dogma. So basically, what is the central dogma? Central dogma is basically the flow of information. As we know that the information is flowing from the DNA to the RNA, that same information is flowing from the RNA to the proteins. Alright, so that flow of information is what we call the central dogma. Alright, so here we have two steps which are involved in the central dogma. Wait, let me write here. So first step is transcription and another step is translation. Alright, so wait, let me write here. As we know that DNA can give rise to RNA. Wait, let me write here. DNA can give rise to RNA. Alright, so when DNA is giving rise to RNA, so basically this process is known as transcription. Alright, so now what is translation? When RNA is forming proteins. Alright, wait, let me write here. When RNA is forming proteins. So basically this process is known as translation. When DNA makes RNA, this process is known as transcription. When RNA makes proteins, so this process is known as translation. So today we are going with the process which is transcription. Alright, we'll be forming RNA from the DNA today. Okay, let me scroll down. Okay, so now basically this we have RNA. Let's suppose, and we know that in RNA what do we have? Genes. Alright, we have genes. So basically this is a gene. Alright, now we need to know that is whole DNA is responsible for making RNA? No, not at all. Only 1 to 2% of DNA makes RNA. Alright, so here we have gene which is a structural gene. Alright, so here we have 5' end, not 5, but here I'm going to write 3' end, and here I have 5' end. Alright, this is 5' end. So if this is 3' end, then here would be 5' end, because this is the complementary strand of this, and here is 5, so here would be 3. Alright, so this is, we have structural gene. Wait, let me write here. Structural. This we have structural gene. Alright. Let me mark here. This is the structural gene. Alright. Now we are going with the formation of RNA. So for forming the RNA, we have an enzyme. Alright, so that enzyme is known as DNA dependent RNA polymerase. Why that is known as DNA dependent RNA polymerase? Because that enzyme is dependent on DNA. As we know that RNA is formed by DNA. Alright, so that enzyme is dependent on DNA. So that's why we have a name that is DNA dependent RNA polymerase. So in human we have, not in human but in eukaryotes, we have 3 types of RNA polymerase. So wait, let me write here. Types of RNA polymerase. Alright, so we have here 3 types of RNA polymerase. Types of RNA polymerase. So here I'm going to write. So first of all we have RNA polymerase 1. We have RNA polymerase here we have RNA polymerase 1. Wait, let me write here. This is RNA polymerase 1. Alright. So second we have RNA polymerase 2. Then third we do have RNA polymerase 3. Alright. RNA polymerase 3. So now let's see what these RNA are making. Alright. So this RNA polymerase 1 is making rRNA. RNA. Alright. This RNA polymerase 2 is making mRNA. This is mRNA. And this RNA polymerase 3 is making tRNA. This is tRNA. Alright. And here I am going to mention types of RNA. Alright. So here we have types of RNA. So we have three types of RNA. rRNA, which is ribosomal RNA. Then mRNA, which is messenger RNA. Then we have tRNA, which is transporter RNA. Alright, so we have these three types of RNA. Now, this is very important to know that what type of RNA we are having in the eukaryotes. Alright, so in rRNA, we have three furthermore types. First is 28S rRNA. Second, we do have, this is the first one. Second, we do have, which is 18S RNA. This is 18S RNA. And third, we have, that is 5.8S RNA. 5.8S RNA. So these were the types of rRNA, which is ribosomal rRNA. Alright. So now let me tell you about the mRNA. So mRNA is not made like this. Like in bacteria, mRNA is made very simply. Alright. So here in eukaryotes, this won't be made very simply. But here instead, hRNA will be made. This is hRNA. Then this hRNA will be modified and will be converted into this mRNA. Alright. So now let me go towards the tRNA. So in tRNA we do have types as well. So in tRNA, the first we have, that is 5S RNA. Then we do have snRNA. We have these two types in the tRNA. Alright. Okay, so now we are going towards the process transcription, like how RNA is made from the DNA. Alright, as I told you, this is the structural gene we have, and this is we have RNA polymerase 2. Because I am going to make here the mRNA first. As we know that mRNA will not be made, but instead hRNA will be made. Then we will be modifying hRNA and we will be converting that hRNA into mRNA. Alright. So here I have taken R2, which is RNA polymerase 2, because we know that RNA polymerase 2 will be forming what? Will be forming mRNA. First of all, it will be forming hRNA. Alright. So to initiate this, we have a sequence here. Like as we have door in the house to enter the house. Alright. So this is the door of the entrance of the house. Alright. So this door is what? This door is promoter sequence. This is promoter sequence, in which we will be entering, not we, but the RNA polymerase will be entering. So for suppose RNA is here now. Alright. RNA 2 is here. Now let me tell you the position of this promoter sequence. So first of all, we need to give the names. Like if promoter sequence is on the 3' end, so that 3' end would be what? Would be the template strand. So let me write here. It would be what? It would be template strand. If this is template strand, then this would not be template strand. We know that. So this would be what? This would be non-template strand. Non-template. Alright? But here is an interesting thing. Here it is non-template strand, but here it has got any other name too, and that name is again we do have, which is coding strand. We know that it is not coding for anything, but still it has gotten the name which is coding strand. Alright? So wait, let me mark here. This is coding. And if this is coding strand, then that would be non-coding strand. Alright? This is then non-coding strand. Wait, let me write here. This is non-coding strand. Thirdly, it has another name too, which is sense strand. If this is sense strand, then it would be anti-sense strand. Alright, so I'm not going to write this now. So we have the position of promoter sequence if we are going according to the template strand. Alright, so promoter sequence is always on the 3' end of the template strand. Promoter sequence is always on the 3' end of the template strand, but we'll be not going according to the template strand. Alright, we'll be going to according to the structural gene. Alright, so promoter sequence is always on the 5' end of the structural gene. We would be not going here according to the template, but instead we'll be going here with the according to the structural gene. Alright, so promoter sequence is always present on the 5' end of the structural gene. And here we have terminator sequence. Terminator sequence. Wait, let me write here. Terminator sequence. And terminator sequence is always on the 3' end of the structural gene. Alright, this is 3' strand, and terminator sequence is always on the 3' end of the structural gene. Alright, now this RNA polymerase 2 is here. What it will do now? It will start making the nucleotides. Alright, for suppose it is making a strand. As we know that RNA is single strand. Alright, if the both gene will make, if template and non-template will make the RNAs, then there would be more than type of RNA. Alright, if there would be more types of RNAs, that means that would not form protein, because that would be complementary strand of the RNA. That would be double stranded. Alright, so we know that double stranded RNA would not make protein at all. Alright, so for suppose if I have a sequence here which is A, here I am going to write A, T, U, not U, but it's also A, C, T. Alright, so this would be going further. So here RNA polymerase 2 will be making nucleotides. Alright, if A is there, we know that the major difference between RNA and DNA is what? Is the first of all, the difference is of oxygen, because there in the deoxyribonucleic acid, oxygen is absent on the second position, while in RNA oxygen is present. But the second difference is what? Second difference is in the nucleotides. Alright, all nucleotides are the same, but only one is changed. Instead of thymine in the RNA, we would be replacing thymine with the uracil. Alright, so if A is there, there would be T, but we know that T would be replaced with what? T would be replaced with U. Alright, so here is T, here would A come. G is there, C will come. If C is there, then G will come. Alright, T is there, so adenine will come. So it will make further nucleotides. There, all nucleotides will be made. Alright, so finally RNA polymerase 2 has reached here now. Now here it has to go back. So from where it would be going back? Alright, it would be going back from this terminator sequence. Alright, so from here RNA will come back. RNA polymerase 2 will come back. Now what will happen? As we have taken RNA polymerase 2, which means we are going to form the hRNA first. So here hRNA is formed. Alright, this is not mRNA because I have told you hRNA will be modified, then it would be converted into mRNA. Alright, so here we have hRNA which is made, and now it would fall down. Alright, this is what? This is hRNA. This is what? This is hRNA. Now we know that hRNA has to be modified for converting this into mRNA, into messenger RNA. Alright, so now we are going towards the processes which are involved in converting this hRNA into mRNA. Alright, so now I'm just going to erase this little bit so that I can write here. Alright, so here I am going with. So first of all, how many processes are involved in converting this hRNA into mRNA? Alright, let me write here the steps which are involved in conversion of hRNA into mRNA. Alright, so what those processes are, basically the first is keeping. Alright, then the second is we have, which is peeling. Last but not the least, we have slicing. Alright, so we have these three processes which are involved in the conversion of this hRNA into mRNA. Alright, so let me use this. There are three steps which are involved in the conversion of hRNA into mRNA. So the first step is capping. Let's see capping. So basically think that this is what? This is hRNA. Wait, let me make it more clear. With the blue one. Alright. Let me make it straight line. So here we have hRNA. Alright. This we are going to just convert it into mRNA. Alright. So basically, for suppose here are nucleotides. But this is, we know that this isn't mRNA yet. This is hRNA, which means it does not contain any type of information into it. Alright, so first step is what? First step is capping. So now we have to make it wear a cap first. So that cap will be made of what? Let's see. So that cap will be made of, wait, let me write here. That would be of methyl gyno tri phosphate. Alright, so this cape is made up of methyl gynotriphosphate. Wait, let me make the structure here so that it can be more clear to you guys. For suppose this is a structure. Here we have gynosine, which is a nucleotide, alright. And as it is a triphosphate, which means here we have one phosphate, then again here we have second phosphate, again we have third phosphate, and here we have methyl with the gynosine. Alright, so this is basically structure of methyl gynosine triphosphate. So here I'm just going to make a cape. So basically this is a cape. Alright, so caping is done. Caping is done of what? Methyl gynosine triphosphate. Alright, this is methyl gynosine triphosphate cape. Alright, now the second process is what? Second process is tailing. So tailing will be done of what? Let's see. Tailing will be done of adenylate acid. Wait, let me write here. Adenylate. Alright. So adenylatic acid. Alright. So here I'm just going to make the structure of this. For suppose this we have a structure of this acid. Here we have adenine, which is a nucleotide, and here we have only one phosphate group. Alright, so this is the structure of this. And here the caping will be done of this adenylatic acid. Alright, so here I'm just going to make the tail of this. So basically this is the tail, and here an important thing is that only one adenylatic acid is not required, but here would be 200 to 300. The range is what? 200 to 300 adenylatic acids are required to make the tail. Alright, so let me tell you where it would be. So suppose one adenylatic acid is here, another is here, another is here, and other is here, and other is here. So we require 200 to 300 adenylatic acids for making this tail. Alright, so the second process is done too. Now the very important process is what? It is slicing. Alright, now we have to cut the unimportant parts. Alright, so now for suppose this is coding for something, again this is coding for something, this is coding for something, this too, and this as well. Now splicing will be done of the parts which don't form protein. Like this would be forming protein, this would be forming protein, this would be forming protein, this would be forming protein as well. But now these parts won't form protein. So we have to do slicing of these parts which are not forming protein. Alright, so the parts which are forming proteins are known as what? These are known as exons. This is exon, this we do have exon, again this we do have exon. And the parts which are not forming exons, which are not forming the proteins, are known as what? These are known as introns. Alright, so these are introns. Introns are what? Introns are the parts of the RNA which are not forming protein, and exons are the part of RNA which are forming protein. So these are basically what? Exons and introns. Alright, so now what we have to do? Let's see. This would be coming up now. Alright, these parts which are not forming the proteins. Alright, Here we have again exon. Again this intron will go up. Again we do have exon here. Again this part will go up. Alright, this would be happening. So now what we have to do here, we have spilosome, which is made of intron, snRNA, which is the part of tRNA, transporter RNA, and protein would be there. Alright, so these will be cut off. We have to cut these. Alright, here I am removing. I have removed this. I have removed this. Now what are left? Let's see what things are left. Now only the parts are remaining which can form the protein. Alright, means only exons are remaining. So now we have to combine these all exons. Alright, so let me erase it and make it again. Wait, let me select it. So now erasing it. I'm going to just make it again. Alright. To make it again here. This we did keeping we did with what? We did with the methyl gynosine triphosphate. Alright. Then we did tailing. Tailing we did with what? Adenylatic acid, which are required 200 to 300 for make tailing for the mRNA. Alright, so now we have to combine these all exons together because we have now cut these introns. Alright, snRNA was there which cut all the introns. So introns were what? Introns were the part of RNA which were not forming protein. Alright, now we have to combine these all exons. Alright, so I'm just going to select any other marker. Okay. So now these all would be combined together. These all would be combined together, and now this would be known as mRNA, which is messenger RNA. But this happens only in the case of eukaryotes. What happens in the case of bacteria? Like bacteria does not need modification. Like there only one RNA polymerase works for forming all those three RNAs. Alright, rRNA, mRNA and tRNA. These three are formed by only one enzyme, which is RNA polymerase. Alright, there are not types of RNA polymerases, but instead there is only one RNA polymerase, and that same RNA polymerase is forming rRNA, mRNA and tRNA. And there we don't need modification because there is directly mRNA is forming instead of hRNA. But in eukaryotes, hRNA is made instead of mRNA. Then that hRNA is converted into mRNA by these three steps, which are capping, tailing and slicing. Capping I told you, which is done with the methyl gynosine triphosphate, which is here as you can see. Alright. So second step was tailing. Here you can see the tail, which was done with the adenylatic acid, which are required 200 to 300 for making tail. Alright. So thirdly we need to splice it. Okay. So these were the parts which were forming the protein, and these were the parts which were not forming the proteins. Alright. So these parts were known as introns, which were not forming protein, and the parts which were forming the proteins were known as exons. So we need to cut these introns. Alright. So for cutting the introns, we were having an enzyme which was snRNA. So snRNA did what? It cut whole the introns. Alright. So now only the exons were remaining. Then we combine these all exons, and now basically the mRNA is formed. So this was the process how mRNA is formed. So now let's see. As we know that, so we know that RNA polymerase. I am not writing polymerase, but instead I am writing P. Alright. One is forming what? It is making rRNA. rRNA, which is ribosomal RNA. Then RNA polymerase 2 is forming what? HRNA. Then that HRNA is converted into what? mRNA. Alright, as we did the processes. Alright, mRNA, which is the messenger RNA. And last, which was the RNA polymerase 3. It was forming what? It was forming tRNA, which is transporter RNA. Now let's see, these are the same processes. Like if we have done one, so these will be made with the same process. Just mRNA was not made. Just because HRNA was made, then we modified, then mRNA was made. Alright. So RNA polymerase 1 will be taking, then rRNA will be formed with the same process. HRNA was formed. Alright. So when we will be taking RNA polymerase 3, then tRNA will be formed, which is transporter RNA. Now let's see what is their work and where they are present. So let me tell you a thing, that transcription happens in what? Transcription happens in what component of cell? Alright, it happened in the nucleus of the cell. Alright, because we know that DNA is present where? DNA is present in the nucleus of a cell. Alright, so from DNA RNA is formed. Alright, so from DNA RNA is formed in what component of the cell? It is formed in the nucleus of the cell. Alright, so now let me make here. For suppose this is a cell we do have, this is a cell we do have, and this is a nucleus. Alright, this is a nucleus. This is a RNA. From here mRNA was made. Alright, this is what? This is mRNA. Alright, this mRNA was made in what? As you can see here, this is the nucleus of the cell. Alright, and this is the cell. So transcription happened in the nucleus of the cell, while translation will be happening in the cytoplasm. Alright, so as you can see, if we are going with an example, let's suppose rRNA is already made. Alright, rRNA is already made and it has came to the cytoplasm. Alright, this is the burger like structure, which are ribosomes, and these are made by rRNA, ribosomal RNA. Alright, and here we have another type of RNA, which is transporter RNA, that is also made, and that is in cytoplasm. Alright, so basically this we do have, that is transporter RNA. Alright, so here translation will be happened, in which RNA will be giving rise to the proteins. That we'll be doing in the next video. Here we have done the process which is transcription. Alright, which happened in the nucleus of the cell. If we do talk about the bacteria, like where transcription and translations are happening there in the bacteria. So transcription and translation are happening in the same component of the bacteria. Alright, in the same component there, because we know that bacteria are what? Bacteria are the prokaryotes. They don't have proper nucleus. So here, for suppose if mRNA is made, alright, from the DNA in the cytoplasm, alright, then here the protein will be made in the cytoplasm as well. So transcription and translation both are happening in the same component in the cytoplasm. But talking about eukaryotes, transcription is happening in nucleus, while translation is happening where? It is happening in the cytoplasm. Alright, so in next video we'll be going with the translation. Here we are done with the transcription. Alright, so bye bye.